Good morning everyone. In the last lecture, we have gone through syllabus of Pharmaceutical Analysis 1 as described by PCI, which is of 45 hours and which is divided into 5 different units. That is Unit 1, which comprises of introductory part of pharmaceutical analysis, and number of hours allotted for Unit 1 is 10 hours. In Unit 2nd, two chapters, that is acid base titration and non-aqueous titration, has been given and it is also 10 hours. The third unit comprises of chapter that is precipitation titration, complexometric titration, gravimetric and diazotization titration, and number of hours allotted for unit 3rd is also 10 hours. So unit 4 comprises of redox titration and number of hours allotted for redox titration is 8 hours. In unit 5 we are going to cover up various electrochemical methods of analysis, say for example conductometry, potentiometry and polarography, and number of hours allotted for unit 5 is 7 hours. So in today's lecture, we are going to start up with the first unit of Pharmaceutical Analysis 1, that is introductory part. So first unit of Pharmaceutical Analysis 1 comprises of definition of analysis, scope of analysis, different techniques of analysis, methods of expressing concentration, what is primary standard substance, what is secondary standard substance, method and standardization procedure of various molar and normal solutions, what are errors, what is pharmacopoeia, what are impurities and limit test. These are the bits to be covered up under unit first of pharmaceutical analysis 1. Of this unit first, today we are going to cover up with the definition part and classification part of analysis. So before starting up with the classification of pharmaceutical analysis, we should know what is analysis. So analysis, the name itself indicate analysis is nothing but finding out either composition of the drug substance or concentration of the drug substance. So analysis is an application of process or a series of process. With the help of a process or series of process, we can be able to identify the given unknown compound, or we can be able to quantify, in that means in what amount the drug substance is present. We can be able to analyze the components of solution or a mixture, or determination of structure of chemical compound which is unknown to us. So in analytical chemistry, mainly we are going to deal up with either identification of given unknown compound or either quantification of given unknown compound. Depending upon this definition of analysis, we can be able to classify the analysis broadly into two major classes, that is qualitative analysis and quantitative analysis. So firstly, we are going to see what is qualitative analysis. Qualitative analysis briefly gives the information about the atomic and molecular species or the functional groups which are present in unknown sample. So it gives the presence of atom or presence of molecule in an chemical entity or in a drug substance. Say for example, with the help of boiling point, with the help of melting point, with the help of either specific gravity, IR, absorbance or optical rotation, viscosity or particle size, we can be able to identify the given unknown drug substance, that is which atoms or which molecules are present in it. Next major class of analytical chemistry is the quantitative analysis. It gives detailed information about the relative amount of one or more of the analyte in numerical terms. That is, in quantitative analysis, we are going to find out the exact either amount, either concentration, or either percentage of the drug substance or an analyte present in it. So majorly qualitative analysis is just the identification of atoms or molecules present in an unknown drug substance or unknown given sample. But in case of quantitative analysis, we are going to find out the exact amount either in the form of percentage, either in the form of gram, or either in the form of kilogram, or either in the form of concentration. So these are the main two classes of analytical chemistry, that is qualitative analysis and quantitative analysis. The whole syllabus of pharmaceutical analysis 1 is of quantitative analysis 1. That is, we are going to determine the concentration or amount of drug substance present. So this analytical chemistry, that is quantitative analytical chemistry, is again subdivided into four major types, that is chemical method, physicochemical method, microbiological method and biological method. Of this, we are going to study in detail chemical method of analysis, which is distributed into five different units of your pharmaceutical analysis 1 syllabus. So this is the major four classes of quantitative method of analysis. Chemical method, that is with the help of chemical reaction that occurs, we can be able to find out the concentration or amount of analyte. Next is the physicochemical method, the chemical reaction which occurs that can be measured with the help of a specific physical property, that is which is also known as instrumental method of analysis. Next major type of quantitative analysis is the microbiological method and biological method. So depending upon the nature of chemical reaction occurred in an unknown drug substance or in an unknown given sample, this chemical method of analysis is again categorized into three main classes, that is volumetric analysis, gravimetric analysis and gasometric analysis. Volumetric analysis is also known as titrimetric analysis. So firstly we will study the definition of volumetric analysis. So in volumetric analysis, with the help of volume of standard solution consumed by an analyte, we can be able to find out the concentration of analyte, particularly various standardization procedure and assay procedure that comes under volumetric types of analysis. Depending upon the volume of standard solution consumed by an analyte, we can be able to find out the concentration of analyte. Next major part of chemical method of analysis is the gravimetric analysis. Gravimetric analysis is an advanced or higher form of precipitation type of titration. In gravimetric analysis, majorly we are going to convert the annihilate into precipitate form. We are going to convert the precipitate into appropriate weighable form, and from the weight of the precipitate and from some basic calculation, we can be able to find out the concentration of annihilate. The results given by gravimetric analysis are more accurate as compared to volumetric type of analysis, but it is more tedious one as compared to volumetric one. Third major class of chemical method of analysis is the gasometric analysis. The name itself indicates from the measurement of volume of gas consumed by an analyte, we can be able to find out the concentration of analyte. So these are the three main classes of chemical method of analysis. What is chemical method of analysis? Depending upon the chemical reaction occurred between the reactant and product, we can be able to find out the concentration of analyte. So chemical method of analysis is again further classified into three major classes, that is volumetric analysis, gravimetric analysis and gasometric analysis. Volumetric analysis is also known as titrimetric analysis. Majorly different titrations are performed, and depending upon the nature of analyte, this volumetric analysis is further classified into five different classes. That is either neutralization titration, non-aqueous titration, precipitation titration, redox titration or complexometric titration. So in volumetric analysis, from the volume of the standard solution consumed by an analyte, we can be able to find out the concentration of analyte. And if the nature of analyte is nothing but the substance whose concentration is to be find out. So if the nature of analyte is acidic or basic in nature and the analyte is soluble in water, then we are going to perform acid-base type of volumetric titration. That is the first class of volumetric analysis. And if the nature of analyte it is weaker one, that is if it is weak acid or weak base one, at that point with the help of with the help of some organic solvent, we are going to dissolve the drug substance, and we are going to convert the drug substance or analyte into specific plus and minus form, that is cationic and anionic form, and we can be able to find out the concentration of the analyte. So it comes under non-aqueous type of titration. So preferably non-aqueous type of titration is to be carried out for the drug substances that are insoluble in water. Third type of volumetric type of analysis is the precipitation titration. If the drug substance or the analyte contains majorly halides in it, say for example halide in the form of either chlorine, bromine or iodine, for that type of analyte we are going to perform precipitation type of volumetric titration. In precipitation type of titration, a standard solution of sodium nitrite is used, silver nitrite is used. Fourth type of volumetric type of analysis is the redox type of titration. If the analyte whose concentration is to be find out, if it is undergoes oxidation or reduction type of reaction, then we are going to perform redox titration, and from the volume of the standard solution consumed by the analyte we can be able to find out the concentration of analyte. Last type of volumetric analysis is the complexometric type of titration. If the analyte or drug substance contains metal ion in it, metal ion either in the form of calcium, magnesium or aluminum, can be determined by complexometric type of titration. Here in complexometric type of titration, we are going to treat the drug substance containing metal ions with an appropriate complexing agent, and formation of complex between metal ion and standard complexing agent takes place. And the standard solution majorly used in case of complexometric type of titration is the disodium EDTA. So this is the classification of chemical method of analysis, that is volumetric, gravimetric, gasometric, depending upon the nature of analyte, whether it is acidic, whether it is weak acidic, whether it contains halide, whether it undergoes oxidation reduction, or whether it contains the metal ion in it. Volumetric type of analysis is categorized into neutralization titration, non-aqueous titration, precipitation titration, redox titration, and complexometric titration. We are going to cover up these five major types of volumetric analysis in pharmaceutical analysis, one syllabus, which is majorly distributed into five different units. प्रत्येक टायट्रेशनच्या अंडर काही स्टँडर्ड सोल्युशन्स येतील आणि काही ड्रग सबस्टन्स येतील. त्या स्टँडर्ड सोल्युशनचं स्टँडर्डायझेशन प्रोसिजर असेल आणि त्या ड्रग सबस्टन्सचं त्याच्या अंडर येणाऱ्या ड्रग सबस्टन्सचं असे प्रोसिजर असेल. that we are going to study stepwise in the next unit. So this is the major classification of chemical method of analysis. Next is the physico-chemical method of analysis, which is also known as instrumental method of analysis. So instrumental method of analysis is based on the relation between the content and corresponding physico-chemical and physical properties of chemical substance being to be analyzed. The change in the physical property of the system are generally detected with the help of detected through the measurement of either current, measurement of either potential, measurement of either electrical conductivity, measurement of either optical activity, refractive index, or with an specific suitable sensitive instrument, depending upon which physical property we are going to measure, whether it is potential, conductance, current, radiation. Depending upon this physical property, a specific sensitive instrument is used, and with the help of instrument and which physical change occurs in chemical reaction, we can be able to find out the concentration or amount of analyte present in given sample. So this is the second major part of quantitative analysis, which comprises of our fifth unit of pharmaceutical analysis 1. Of this we are going to study in detail. Instrumental part मध्ये in detail येईल conductometry, potentiometry and polarography. मग प्रत्येक इन्स्ट्रुमेंटचा प्रिन्सिपल असेल, वर्किंग प्रोसिजर असणार आहे आणि अप्लिकेशन पार्ट असेल. So this is the second major class of quantitative analysis that we are going to study in fifth unit of pharmaceutical analysis 1. The results given by instrumental part of analysis are more accurate as compared to older volumetric, gravimetric or gasometric one. So present day of instrumental analysis gives better and faster result as compared to the older one. The data given by the instrumental part of analysis are more accurate and we can interpret the data either in the form of images or either in the form of graph, and we can be able to find out its concentration. Miniaturization can also be done with the help of instrumental method of analysis, and the data which is obtained, that is better data processing, can be done by instrumental method of analysis as compared to the classical method of analysis. So these are the advantages of instrumental method of analysis as compared to classical one. That is, it gives better and faster result. More data can be obtained from instrumental method of analysis, which can be converted into various images or graphical form, and easily we can interpret the result. Next is miniaturization is also possible in instrumental method of analysis, and data processing method gives the better result as compared to other one. Last bit related to your introductory part of analysis is the importance of pharmaceutical analysis. Analysis is important in every step of manufacturing of either customer product or manufacturing of either pharmaceutical product. If we are dealing with the manufacturing of pharmaceutical product, the manufactured product should be highly potent, safe and effective one. So analysis is very important in every step of manufacturing, that is right from identification of raw material, checking the quality of the drug or product during in process and also during the finished products. We can be able to analyze the drug products at three major steps of manufacturing, that is at the raw material. Also we can be able to analyze the drug substance during processing. Also we can be able to analyze the drug substance whether it is highly pure or not. Nextly we can able to analyze the drug substance at finished product steps, so that impurity is will not get incorporated into the final product. Next is analysis also plays a very important role in determination of additional impurities. Impurities are present in any sort of the drug, in any sort of the drug products. Impurity either in the form of chlorine, bromine or iodine that can be incorporated. So analysis helps in finding out the impurities and minimizing its concentration into the sample. Nextly, analysis with the help of analysis, we can be able to find out the stability of the drug products by analyzing it at various steps, that is at raw material, identification of raw material, in process and finished one also. Next is analysis plays a major role in determining the strength and concentration of chemical compounds by determining either the standardization procedure or by doing either the assay procedure. We can be able to determine the strength as well as we can be able to find out the percentage of the drug substance. Next, analysis also helps in determination of the molecular weight of the chemical compound. With the help of analysis, we can be able to find out molecular weight, we can be able to find out the structural elucidation of the synthetic compounds, that is which molecules or which atoms are present in a given unknown sample, we can be able to find out. Next is we can able to find out the concentration of the drug substance in plasma or in biological fluids by performing the analysis. Means how much drugs has been released from the given dosage form can also be calculated by doing an analysis. Next is the determination of pK values, partition coefficient, solubilities and stability of drugs can be done, of the drugs under development can be done with the help of analysis. So these are the application or importance of pharmaceutical analysis in day-to-day life. With this, we have completed the introductory part of pharmaceutical analysis, that is what is analysis, what are the types of analysis, uh, of the major classification of analysis. We are going to cover up the chemical method of analysis. Next is the instrumental method of analysis, how instrumental method of analysis is gives better result as compared to classical method of analysis, and last is the importance of pharmaceutical analysis. In the next lecture, we are going to study various methods which are used for expressing the concentration of solution.